In this mini lecture, I'm going to go over bacterial protein expression. Now, any biochemist that wants to work with purified proteins is typically going to have to express that protein in some other organism. It is possible to do this in a eukaryotic expression system, but by far the most used system for producing proteins is in bacteria, and that tends to be in E. coli. We are going to do that in our class as well. So what I'm going to do in this lecture is go over the strategies in particular for inducing expression of your protein of interest in a bacteria. The typical flow path for purifying proteins is the first thing to do is to construct a plasmid. And that I'm going to show you a plasmid in just a second. The key thing there is that the plasmids must have the proper elements so that a gene of interest can be produced by the bacteria. Then you're going to transform that plasmid into a bacterial strain and then do a process called protein induction. And that's what I'm going to spend a fair bit of today's mini lecture on. And then once you have the bacteria produce your protein of interest, you go on to purify your protein. And this is what we'll spend the latter part of our class working on. So this is a common plasmid system that is used for producing proteins in bacteria. And these vectors are called PET vectors, P-E-T. There's a family of these, and PET15 is a very good representative of what this class of plasmids looks like. And I very commonly use PET15B for production of proteins of interest in bacteria. So as for all plasmids that we use in molecular biology, they're going to have an origin of replication, which is represented here by ORI. They're going to have some antibiotic resistance marker. In this case, this is a gene that encodes for resistance to ampicillin. You will always want to pay attention to what the antibiotic resistance you're using is. And then there's going to be what's typically called a multiple cloning site into which you would put your gene of interest. And for example, if you wanted to express some other protein in E. coli, you would clone your protein of interest into this multiple cloning site, this MCS region. So let me zoom in on this region because this is sort of where a lot of the business of what we're going to be talking about occurs. So this is that multiple cloning site, and there's a number of elements that are on this site. And so typically, these are all these little numbers, these little letters up here, for example, NDE1, XHO1. These are restriction enzyme sites, which allows you to linearize or open up your plasmid of interest. And you would clone your gene of interest. And for PET15B, we typically clone into this NDE1 site. That ATG is going to be the start methionine, and your gene would end up being put in frame just downstream of that. There are a number of elements up here that are important for expression of proteins in bacteria. So there's a number of things I want to sort of pay attention to. One is there is a promoter sequence, and notice that this says T7. So T7 refers to T7 bacteriophage. And so this is a promoter sequence that gets recognized by the T7 phage RNA polymerase. So the RNA polymerase produced by the T7 phage would bind to this sequence, and then it's going to start transcribing sequences downstream of that promoter element. This is an important consideration because since we're using a phage promoter, we need to make sure that we've got a source of the T7 RNA polymerase. So I'm going to get to that in a bit. There's this LAC operator sequence that's downstream of that. This is also important because this DNA sequence is bound by the LAC repressor. And we're going to take advantage of this as a way of regulating our expression. So this is going to be a very important element for what we're going to talk about. Notice my note down here that you want to pay attention to your transcription start site and your translation start site. So the transcription start site is going to be just downstream of this T7 RNA polymerase gene. And then notice the sequence right here, which is a ribosome binding site. Once we have our messenger RNA that's been produced by transcription, the ribosome in bacteria is going to bind to the transcript and scan down until it finds this RBS, this ribosome binding sequence, and then it's going to start translating at this ATG right here. Okay, so this is your translation start site. as with all translation start sites, it's an ATG, and importantly, it sets the frame, meaning that the ribosome is going to scan down every 3 nucleotides and insert the proper amino acid into the growing polypeptide chain. Okay. Notice that on this particular sequence, we have a bunch of information that's going to be upstream of our ND1 site. This thing here is a hist tag. This is a sequence of 6 histidines. And in the protein that gets produced in the bacteria, we're going to take advantage of these histidines as a purification strategy. There is an affinity purification strategy in which histidines bind to divalent metal ions. And that's something we're going to use in that sort of third phase that I'm not going to get into much more in this lecture here. One more sequence here is this T7 terminator. This is a transcriptional terminator sequence that's going to be in here so that the RNA polymerase knows when to stop transcribing its genes of interest. So just to reiterate this point, in constructing your plasmid, you would insert your gene of interest into this sequence right here. You would likely want a stop codon at the end of that sequence, but what you would end up having in the resulting protein would be this 6 histidine tag that would be on the N-terminus of the resulting protein. So I want to go over some of the elements with the lac operon because this is how we're going to end up regulating induction of our protein of interest. And recall in that previous slide that I just showed you that there is a lac operon DNA sequence just downstream of the T7 RNA polymer, the T7 promoter. Let me just show you that right here. So this lac operator sequence right here is downstream of the T7 promoter sequence, and this lac operator sequence is going to basically block transcription from the T7 promoter unless we do something to remove the lac repressor. So here are the elements that are present within the lac operon. So the lac operon consists of 3 genes that are involved in metabolism of lactose. So this refers to LAC-Z, LAC-Y, and LAC-A. I'm going to refer to this gene later on. It's the lactose permease is going to be important for bringing in lactose from the outside to the inside of the bacteria. And then beta-galactosidase actually has an important role in converting lactose into another molecule called allolactose. I'll show you what these look like in just a second. But from the bacteria's perspective, what they've evolved to do is they do not want to have to be producing any unnecessary genes unless those genes are required. And so if a bacteria doesn't have lactose to metabolize, it essentially wants to keep these genes from being transcribed and translated because it's energetically costly. And so they have this regulatory system that's up here, which includes the lac operon, or the lac operator. So the way that what happens with the lac operator is it's a DNA sequence that gets bound by the lac repressor. So the lac repressor is this protein right here that's encoded by the LAC-I gene. And the lac repressor can bind to the lac operator and keep transcription from this upstream promoter off. Okay. Now it's going to do that in the absence of lactose. Now in the presence of lactose, what's going to happen is a little bit of lactose will get into the cell. This LAC-C gene product, the beta-galactosidase, will convert lactose to allolactose. Allolactose will then bind to the lac repressor, and it's going to cause that lac repressor to fall off DNA. That essentially frees up RNA polymerase to start transcribing genes downstream of this. So here are some notes. This allolactose is the true inducer of this system in that it's the molecule that binds to the lac repressor and prevents binding of the lac repressor to this lac operator DNA sequence. CAP is a transcriptional regulator that positively regulates transcription, and it is allosterically regulated by cyclic AMP and also negatively regulated by glucose. And a key point which I alluded to earlier is that E. coli conserves cellular resources and energy by not making these 3 lac proteins when there's no need to metabolize lactose, such as when other sugars like glucose are available. So I want to go over the sugars that are involved in regulating the lac repressor. So this is lactose, that's right here, which can be cleaved down to galactose and glucose. These sugars can then be metabolized by a bacteria. In the previous slide, I talked about this allolactose, which will be converted from lactose into allolactose by the gene product of beta-galactosidase, or the gene product of the LAC-Z gene, which is beta-galactosidase. So this is allolactose. This is that true inducer that will bind to the LAC repressor and cause the LAC repressor not to bind to DNA nearly as well. In the experiments we're going to be doing, or in our protein inductions, we're going to be using this molecule called IPTG, that stands for isopropyl thiogalactopyranoside, and I'm going to be referring to it as IPTG in class. This is an analog of allolactose that binds well to the LAC repressor and has the same effect. In other words, IPTG can bind to the LAC repressor, cause it to fall off that LAC operator. So we're gonna take advantage of this IPTG molecule in our protein inductions. So when you're doing protein inductions, there's a number of obstacles, but I think the biggest two obstacles are shown here. So recall that the goal of our protein expression is to express our proteins to high levels. So we'd like to get a high yield. And the and here is very important. We need to have our protein properly fold. And so what happens when we end up putting a plasmid with our gene of interest into the bacteria is, first of all, you're going to have transcription that's going to make a messenger RNA. That messenger RNA will be read by the ribosome, which is going to synthesize the polypeptide. And then that polypeptide has to properly fold. And that is a process that can very often not go well. And so we pay very close attention to conditions to try and get our proteins properly folded. I just want to bring up this term right here, soluble fraction, because you'll often hear people who do this kind of work frequently will ask you, oh, was your protein in the soluble fraction? What that refers to is when we're purifying our proteins. So this is going to be the downstream step after our protein inductions. What you'll do is we'll take a bacterial pellet, we'll lyse it open in a buffer, and then we end up doing a centrifugation step where we end up pelleting insoluble material. And during that pelleting step, what we want to have happen is our protein to be present in the supernatant, or the soluble fraction. If it's improperly folded, it will partition into that pellet, so it'll be in the insoluble fraction. If it's properly folded, it will be in the supernatant, in the soluble fraction. So if people ever ask you, is your protein in the soluble fraction, really what they're asking is, did it properly fold? So here are two common issues that we encounter when expressing an exogenous protein in E. coli. Number one, your protein is toxic. And there can be a variety of reasons why proteins can be toxic to the bacteria. As an example, if you have an enzyme that's a DNA cleaving enzyme, it's quite possible that that DNA cleaving enzyme will end up cleaving the bacterial chromosome, and the bacteria just won't be able to actually replicate. One thing we'll notice is when we're growing our bacteria that has a protein that we are expressing, sometimes the bacteria grow slowly, or more slowly than when they've got that plasmid than just normally. And that's usually a pretty good indication that they're unhappy with your gene being present, and it's an indication that your gene is toxic. The second thing that we deal with is that your protein doesn't properly fold, and what this results in is what are called inclusion bodies. And so inclusion bodies are these large, or these aggregates of insoluble proteins that form within bacteria, and they can be big enough. When I was saying large, they can be big enough that you can visualize them with a microscope. And so they actually, like under a microscope, they actually look like almost like a small organelle. They do not have a membrane around them, but they have these, they're called sort of bodies, and they're called inclusion bodies. We want to avoid inclusion bodies. And this gets back to what I was talking about earlier. We want our proteins to properly fold. Okay, so we're going to be using the PET system, and these are some of the important components that we're going to need to consider when we're doing our inductions of our bacteria of interest. So there's a lot of stuff going on here, and I want to kind of work through what all these different elements are, because there's a number of very important things to consider here. So first off, on the far right here is a representative PET plasmid, and this is basically what I showed you earlier. And what's being shown here is we would have our gene of interest, our target gene of interest that's been cloned, and then upstream of that is the LAC operator, which is right here, and the LAC operator in this case is being bound by the LAC repressor. So the LAC repressor is binding to that. And then there is going to be the promoter sequence for the T7 RNA polymerase. Now, in this sort of shot right here, the T7 RNA polymerase is being inhibited by binding of the LAC repressor to the LAC operator. So you would have transcription of your gene would be turned off. We also, what's being shown here is the LAC-I gene encodes for the LAC repressor protein. So you need to have a source of the LAC-I gene. We've got that present on our PET plasmids. Now, what's being shown here in purple is a representative E. coli. So this is the host cell. This over here on the left, this purple sort of spaghetti looking thing is the E. coli genome. And there's an important consideration in this strategy in that we need a source of T7 RNA polymerase that's going to be able to bind to the T7 promoter. Recall that the T7 RNA polymerase is a phage gene. It's not a native E. coli gene. So we need to make sure that we're using a strain of E. coli that is producing the T7 RNA polymerase. There are various strategies that you could use for this. The most common is that there is a lysogen called the DE3 lysogen. So a lysogen is a phage-derived sequence of DNA that is incorporated into the E. coli genome. And on the DE3 lysogen, there is this sequence information up here. So there is the gene that encodes for the T7 RNA polymerase. And upstream of that gene is a LAC operator and then a promoter for the native E. coli RNA polymerase. Okay, so the DE3 lysogen it encodes for the T7 RNA polymerase, and this system is also under regulation of the LAC repressor and LAC operator. Okay, so when we're doing a protein induction, we will have transformed into a DE3 containing E. coli, our pet vector, and we will end up growing our bacteria up. And then when we want to turn on transcription of our gene, we're going to add IPTG to the cells. So recall that IPTG is going to bind to the LAC repressor and cause the LAC repressor to fall off of the LAC operator. Okay? So two things occur when we add IPTG. First, over here on the DE3 lysogen, IPTG will bind to this LAC repressor, cause that LAC repressor to fall off. The E. coli RNA polymerase is able to bind to the promoter sequence upstream of the T7 gene, and it is going to end up transcribing the T7 RNA polymerase, which will then make the active T7 RNA polymerase protein. Over on our plasmid, the IPTG will also cause this LAC repressor to fall off of DNA, and now the T7 RNA polymerase protein can bind to its promoter, and it can start transcribing the target gene downstream. One thing I should mention is that the T7 RNA polymerase is a very active RNA polymerase. In other words, it's producing a lot of transcripts, and it's one reason that researchers like to use this polymerase is because it is very active. Let me just review that one more time because this is a really important consideration for what we're doing. We are going to grow our bacteria up to a certain density. We're going to pay attention to that. And when we're ready to turn on transcription of our gene, we're going to add IPTG. IPTG will allow the T7 RNA polymerase to be produced. The T7 RNA polymerase can then bind to its promoter. IPTG will have caused this LAC repressor to fall off of DNA. We get transcription of our target gene. Okay. All right. So I want to point out one more sort of strategy that may come into play, which is, remember how I talked about the fact that sometimes target genes can be toxic, and it's one of the things that we really want to avoid at all costs. And so even though this system looks like it is turned off until we add IPTG, it turns out that the LAC repressor doesn't bind irreversibly to that LAC operator. Instead, what the LAC repressor is doing is it's binding, and with some frequency, it's coming off of the LAC operator, and then it's binding again. And so what that means is you can get a little bit of background transcription of the T7 RNA polymerase, and then you can get a little bit of background transcription of your target gene. Sometimes we refer to this as basal transcription. So it's transcription that occurs without the addition of this IPTG inducer, right? Now the problem with that is that if your gene is toxic, the gene products that are getting produced by that basal transcription can cause your cells to either die or just grow very slowly, right? So in some cases, we want to try and minimize the amount of basal transcription that occurs. one strategy we can use is by adding an additional gene that is encoded by the T7 phage lysozyme gene, and this is going to produce a protein called T7 lysozyme. Now, T7 lysozyme does two things. One is it lyses the bacterial cell wall, but it needs to do this from the outside. So when it's produced inside the cell, it's not going to be lysing the cells. But the second thing that it does is it binds to the T7 RNA polymerase and it inactivates this. So one thing you can do is you can add an additional plasmid. The most common one is called P-lys-S that encodes for the T7 lysozyme. And if you've got this plasmid present in addition to your PET plasmid, you'll have this T7 lysozyme present. It can inactivate the T7 RNA polymerase, and that can help prevent this basal transcription from occurring before you add the IPTG inducer. When you add your IPTG, the key thing is that you want to have enough IPTG so that all of this lac repressor can then be, this lac repressor will end up being removed, and then you can end up having your T7 polymerase being produced. Okay. All right, so here are some elements that I just talked about. Be sure to understand what the DE3 lysogen is, the T7 polymerase, and that P-lys-S plasmid that I encoded for, which encodes for the lysozyme gene, which is a way of keeping down our basal transcription. Okay, so the most commonly used strain of bacteria for protein inductions is what's called BL21. For inductions that use T7 polymerase, which, for example, the PET vectors use, we must have a source of T7 polymerase since it's not an endogenous E. coli gene, and the most common source is that DE3 lysogen. And then some expression strategies will utilize additional plasmids present in the bacteria. I just talked about P-lys-S. And there's another one called PRIL, which encodes for some tRNAs that are rare, that can encode for rarely used codons. I'll mention this in one of the later slides. So the bacterial strains that we use for protein expressions matter, and you're going to want to pay close attention to the bacterial strains that you're choosing to do a protein induction. A typical strain that we would use for protein inductions is going to be BL21, so that's the parental strain. In parentheses, you would see DE3. That's going to be the lysogen that contains our T7 RNA polymerase. And you may have an additional factor such as P-lys-S or PRIL that would be in there. So let me just go through a typical induction that I would do, a typical induction protocol. And this is going to be similar to what we're going to be doing in the class. So first of all, you're going to have a plasmid of interest. I commonly use a PET-based vector, which will often have his tags. There's other vectors as well that can be used. So for example, there's commonly used ones are called PGEX and PMAL that encode for other kinds of affinity tags, and there's others that are out there. But the PET vectors are very commonly used. You're going to want to pay attention to your cells. Since we're using PET vectors, we need to make sure that we've got this DE3 that's present. And then typically we're going to be using a class of media that's called Luria broth. We're going to be adding antibiotics to select for our plasmid of interest, so you need to pay attention to what antibiotic resistance your plasmid encodes for. And then sometimes I'll add a little bit of magnesium sulfate and sometimes glucose, because glucose can end up helping kind of keep down some basal transcription as well. So the first step you would do, which actually isn't shown on here, is we would do a transformation of our plasmid into the bacterial cells of interest, and we would plate them out on antibiotic plates. And that would be sort of day -1 if we're keeping track of how this works. And again, a typical transformation, you have to let your plates grow overnight. So you've got to sort of plan in that extra day. When you're ready to do your induction, here's what you're going to do with your cultures. Day 0, you're going to, in the evening or afternoon, you're going to pick an overnight culture. And I usually use around 12 to 15 mils per 1 liter of culture that I'm going to do the large scale induction for. You'll grow that overnight culture, you'll come in the next morning, and we'll dilute the overnight culture 100-fold into pre-warmed media. And so for example, I would dilute 10 mils from that overnight culture into 1 liter of LB that's got our appropriate antibiotics. We then grow those at 37 degrees, always with shaking, until the optical density at 600 nanometers is 0.6 to 0.8. I typically shoot for 0.6. So this optical density at 600 nanometers is just a measurement of how dense the bacterial culture is. And researchers have basically correlated this optical density with the number of bacteria that are present in the culture. And this is what's going to be present in mid-log phase growth, when the bacteria are doubling basically as fast as they can. And it turns out that that's where optimal protein expression occurs. Okay, so there's some very important things that happen at this OD 0.6, and I pay very close attention to trying to catch the density within this range here. You really don't want to let it go above this. There's an optional choice of putting that culture on ice for 30 minutes, and it turns out I think there's a few things that happen. One is I think you just slow everything down, like the transcription and the translation. And there may also be a feature of inducing a stress response in the bacteria that can include chaperones that may help protein folding. So this is an optional step, and lately I haven't been using it, but traditionally I did use a 30-minute ice step. We are then going to add IPTG to a final concentration of 0.5 millimolar. And we may vary this a little bit, but this is where we're going to basically be adding our inducer. And then we're going to turn down the incubator and shake overnight at 16 degrees Celsius. Okay. The next day we come in, we'll centrifuge the bacteria, and then transfer the pellets to a 50 ml conical tube, and we'll store those pellets at minus 20 degrees Celsius until we're ready to do our protein purification steps. Just some notes on bacterial growth. Bacteria need air to grow. So whenever you grow cultures, make sure to have sufficient headspace of air above the liquid in your tube. The culture volume should be should be at least 1/3 or less the volume of the flask. Okay, so you don't want to have your culture volume taking up more than 1/3 the volume of the flask, is sort of a rule of thumb. Okay, so I want to go over another strategy to induce proteins that's referred to as autoinduction. And this is a variant of what I just showed you with IPTG. So it's going to be a non-IPTG method of induction. And this strategy of autoinduction is based on this property of how bacteria metabolize sugar, which is shown here at the bottom, in this image at the bottom. So if E. coli is grown in the presence of two different sugars, so glucose and lactose, the E. coli will first metabolize glucose. And if you're following the growth, it's going to look like this. You're going to have this phase 1 growth where the bacteria are growing, they're growing, they're growing. And once the bacteria have metabolized all of that sugar, they then have a lag in their growth right here. And what they're doing is they're basically switching over to now metabolizing lactose. And then they start growing again, where they're now metabolizing lactose. And this is going to be in phase 2. Once they've metabolized all those sugars, they'll then go into stationary growth. Okay. So we can take advantage of this feature of how bacteria metabolize sugars to generate what's called autoinduction. So the premise of autoinduction is shown here. So what you're going to do is you're going to grow cells in media that contain glucose and lactose. And you can kind of think of this as glucose as an inhibitor of induction of your protein, and then lactose is going to be your inducer. Okay. So if glucose is present, what's going to happen is catabolite repression and inducer exclusion. So in other words, the bacteria will exclude import of lactose into the cell, will prevent lactose uptake by that lactose permease gene. Okay, so during that phase 1 growth, the bacteria will only metabolize glucose and will exclude lactose from the cells. When the bacteria have reached a certain density, and that certain density is going to depend on how you formulate your media, they will have metabolized all the glucose, and then they'll switch over to lactose. Okay? And so here's what's going to happen when they switch over to lactose. Lactose will be imported by that lactose permease protein. The lactose will then be converted to allolactose by beta-galactosidase. And remember that allolactose is the true inducer. So it's going to bind to the lac repressor and going to start inducing our gene of interest. So the key here is to formulate your media such that you have the right levels of glucose and lactose that meet these requirements up here. And then you want to also have it so that you get optimal growth of the bacteria. This system was generated by a guy named William Studier, and this is actually a worthwhile paper to read, which is sort of the strategy for autoinduction. And I, in my own work, am tending to use the IPTG strategy of induction. I'll tell you though that there are a lot of labs that are commonly that do a lot of protein expressions that are using this strategy of autoinduction. Okay, some other considerations. I want to go over another consideration for when you want to produce an exogenous protein of E. coli. So one of the critical considerations is that different organisms will have different codon usage, and this can affect the expression of exogenous genes in E. coli. So remember that in the codon table, there are the codons that organisms use are degenerate, meaning that most amino acids are encoded by more than one amino acid. And it turns out that organisms have evolved to use for each of the different amino acids, they will use the different codons with different frequencies. And so in the case of E. coli over here on the left, these numbers that I'm showing you are the frequency of the codons that are used by E. coli. And so if we take a look at one of the examples, let's take a look at arginine, which has 6 different codons. You'll see that some of these are used much more frequently than others. So these 2 codons for arginine are used quite frequently, but then there's 1, 2, 3, 4 that are infrequently used by E. coli. Now, if we look at codon usage by Homo sapiens, notice what happens is that the Homo sapiens tend to use these other arginine codons much more frequently. So this creates a situation. Suppose you are going to introduce a human gene into E. coli. One of the problems is that it's quite likely that your human gene is going to be using some of these codons for arginine that are rarely used by E. coli. And it turns out that that can really limit the expression of your protein of interest. Okay, so in that case, suppose you have a gene that contains rarely used codons and you want to express that gene in E. coli. What are things you can do to improve its expression? So I'd say there's really 2 main strategies that you can use. One is you can add a plasmid that contains extra copies of the tRNA genes that are rarely used. And so one of the common used plasmids is called RIL. Remember I talked about that plasmid that has RIL? And what that is the plasmid contains tRNAs that encode for rarely used arginines, isoleucines, and leucines. So the R, the I, and the L stand for those rarely used amino acids. Here's those tRNAs that recognize these various different codons, right? And in fact, I do use this plasmid somewhat frequently, this RAL plasmid, okay? Now, the second strategy is you can codon optimize your gene. Turns out these days, gene synthesis is relatively inexpensive. And I'd say many labs, especially labs that have a fair bit of funding, they don't mess around with cloning their own genes. Instead, what they'll do is they will contract out with what's usually called the CRO, a contract research organization, that will end up cloning their gene of interest into their desired plasmid. And what they'll do is they will codon optimize the gene. What that means is they will have a DNA sequence that encodes for the exact same amino acid sequence, but they'll change the codons so that the codons will be, if there's any rarely used codons, for example, rarely used arginines, they will change those codons to the codons that are more commonly used in E. coli. And if you go with this codon optimization option, you don't need to use the RAL plasmid because you now aren't dealing with those rarely used codons. Okay, I'm going to go ahead and stop here. So we've gone over, just to summarize, we've gone over strategies that we use for protein inductions. As we go through the protein inductions, I think a lot of people kind of look at this and think like, oh, it's not that interesting. You know, we're frankly spending a lot of time making media, growing bacteria up, and then cleaning up after ourselves. But keep in mind that induction of your protein is the most important part of purifying a protein. It is vital that you get large amounts of your protein that's soluble. And if you don't have proper induction conditions, you get low expression levels, and the protein purification that's downstream becomes much, much more difficult.